Hey everybody, welcome back to another episode of Biomolecule Basics with Mr. Coleman. This one, our last one we'll do here, is on proteins, or things more commonly known as polypeptides. So what we're going to take a look at first is some of the basic jobs. The one thing about proteins that is unique to proteins is that they are the most diverse biomolecule of the carbs, lipids, and the proteins, obviously. They have a lot of different jobs because each protein that's created has a specific job. Many of them are involved with different cell parts. As you guys know, proteins are a major portion of, like, muscle fiber is one example. A lot of your major organs are muscles, so all those different types of things are going to have different cell parts or different duties for their cell parts. They're also involved with muscles, like I was saying. They make the fibers that create the contractions in your muscle fibers with the actin and myosin filaments. They are enzymes. This is a picture of a kind of a typical enzyme, which is something that takes and makes reactions happen at a faster rate. Without enzymes, your body would operate but much slower, because enzymes are used to create or make reactions happen much faster. You'll hear more about enzymes in our next unit when we cover sort of cellular biology and how your body deals with different things. So, are going to be involved in transport. Hemoglobin is a classic example. There's a picture of hemoglobin there. Hemoglobin is one of the things in blood that transports oxygen throughout your body. So, there is that. There is also insulin. It's involved in regulation in your body. So insulin or blood sugar levels are also the jobs of proteins, that regulation. And then on my list, they create antibodies for your immune system response. Antibodies are these right here. Oops. They are these Y-shaped proteins right there that are used to fight off infections when you get them inside your body. So all of those are different sort of general jobs that proteins can in fact do. So the next bit gets into kind of the whole structure of them. The first thing I'm going to mention is that they do have a primary, secondary, tertiary, and quaternary structure to them. With the primary structure, proteins are unique in this way. The primary structure is essentially, if you think about it, it is just a chain of amino acids put together. In just one long chain. There can be some minor bends to it, but it's basically one long chain of amino acids. Something like that guy right there. Okay, you can see there's, they list them as A1, A2, amino acid 1, amino acid 2, 3, 4. This one, the example over here, they're a little more specific. The glycine, proline, threonine, glycine again. So that's the code. And whatever that specific order is it determines the function. So primary is a single chain. Then you jump to secondary. Now secondary, you take that chain that you created, and now you can either fold it or sort of coil it to make individual, or make a protein jump to that secondary structure. It's still one primary, but you can see here, you take that primary structure, and now you can fold it, pleated folds they call them, with the alpha helix, that would actually be the coil, the helix shape, or the pleated fold, they call it beta, and that would be the other example. And this is going to be, the folds are held by these hydrogen bonds. In either one of these cases, you're going to see hydrogen bonds, weak hydrogen bonds, that are going to cause the folds to stay or to keep. Now, if you take it one step further, so there's primary, secondary, the tertiary. Now, you're still working with individual chains that have been folded or coiled, but now they are further folded or coiled. So if I jump to the same image, there's primary, secondary structures with the pleated sheet or the helix, and now you get the tertiary structure. So this is folding the already folded things, and then that gives this three-dimensional sort of shape here. This is actually going to build hemoglobin, but you're going to see that what you get is the continuation. You get the hydrogen bonds in there, and then in this one you get some interactions between some of the R groups are going to start to come into play to hold these things together and give them that additional folding. And then the last step, quaternary, forward. This ties it all together from primary to secondary to tertiary to quaternary. And in this case, it gives you take multiple primary structures that have been folded into that tertiary, and then you clump those together. So in this case, you can see here there are multiple subunits. In hemoglobin, there are 4. So there are 4 separate tertiary structures combined into one. So it just shows you that this structure allows for lots of sort of, in proteins especially, structure and function are so closely linked that if you change the structure at all, you change the function of that actual protein. So that's unique to proteins. They have this kind of 4 levels of structure. Just a basic, what are they? We know the monomer is an amino acid. So you take a bunch of monomers and you chain them together to get your polymer, which would be that protein or that polypeptide. Each of those would be a different amino acid bonded by, and there's a specific bond there that you guys should know, and it's called a peptide bond. Alright? And the one thing you need to know, or when you're looking at them, you need to realize that they, pull up a nice picture here, have what's called the amine or the amino group and a carboxyl group. The amine group is the NH2 group, and the carboxyl is the COOH group, the carboxyl or carboxylic acid, which is this side. They also have what's known as an R group right here, and the R group can be one of 20 different things, from a simple H, if it's going to be glycine, to the largest I showed you guys in class was tryptophan, which has a huge sort of 2 carbon ring structure as that R. So they can vary, but your body has need for 20 different amino acids, 12 of which your body can make, 8 of which you must ingest from your food and your diet to break down so that you can reuse those amino acids to create what you need to. So you've got, again, this is one amino acid. This is a single amino acid. Has a amine group, a carboxyl group, that central carbon, and then off of that a single H, and then that R group. They all look the same except this R group is different. To put them together and to take them apart. Well, let's look about putting them together first. You can see here, this is dehydration synthesis to put them together. So this, looking in this direction, is dehydration synthesis. So we have 2 amino acids. Again, you can look and see there's the amine group, there's the carboxyl group. There's another one, amine group, carboxyl group. And what you're going to do between these 2 is you are going to look for the H, the OH, to remove to make water. And you are going to see it's highlighted there in blue. You're going to remove that water, and you're going to have an open bond here and an open bond here, which create this peptide bond right here. So right there you have a protein that's being built. You can call it a dipeptide if you would like, because there's 2 of them. But you're in the works of making a polypeptide through dehydration synthesis. It must be between the OH of the carboxyl and the H of the amine group. Those are the 2 functional groups we're looking at. So that's dehydration synthesis. Take one step forward, and again, you already know. But in this case, I changed it to hydrolysis, so now we are going in this direction. So you add water. So to this one here, you would add water, because again, hydro, water, lyse means to break. And in the addition of water, the OH would add here, H would add here, cuts that peptide bond, and now we have 2 separate amino acids. So hydrolysis would happen, for example, if you ate fish for dinner. Your body digests that fish down into the individual proteins, your body would have to do hydrolysis or add water to break these cells, or break the chain to make individual amino acids available to use to, say, produce muscle fibers or to do whatever job they would be destined for. So those are your two examples: hydrolysis and dehydration synthesis. And that pretty much wraps up all of these different things. We will be testing on this in a couple days, and hopefully you've used these videos to review. Again, you can look on the reading schedule on the website, and you should be able to find the pages that correspond to this in the textbook. And then I also have a couple other things on the website there, review sheet, and some answers to take a look at. There you go. Thanks for listening, and we'll see you in class.